Hello friends, there's an animal that went extinct 13,000 years ago, the dire wolf, and now it has been brought back to life using genetic engineering. This claim is being made by a company called Colossal Biosciences, a company that says it plans to de-extinct many other popular extinct species in the future. Species like the Tasmanian tiger, once found in Australia, the dodo bird, which went extinct in Mauritius in the 1600s, and the woolly mammoth, giant relatives of modern elephants that went extinct around 4,000 years ago. According to the company, the dire wolf is their first successful attempt at de-extincting an animal. The dire wolf species became widely popular because of the TV show Game of Thrones, where many main characters had dire wolves as pets. Inspired by that, the two dire wolves they created were named Romulus and Remus. This entire project was conducted at a secret location in America. In this video, you can see them basking in the sun inside an enclosure. They are now 5 months old. These pups have no idea how unique they are, born into a world where no other members of their species exist. Not only that, a third dire wolf was also born, a female named Khaleesi. At first glance, all of this seems truly amazing, but many people have raised questions. Some experts claim the company's statements are false, saying they haven't actually brought back a dire wolf, but rather genetically engineered a version of an arm wolf, another wolf species. How was all this achieved? Let's understand the whole issue deeply in today's video. The first discovery of direwolf fossils by humans was near a river. In 1858, American scientist Joseph Leidy suggested placing this animal under the Canis genus, giving it the scientific name Canis dirus, meaning a fearsome dog. The Canis genus includes dog-like animals like dogs, wolves, and jackals. Later, in 1918, scientist John Merriam, while studying direwolf fossils at the La Brea tar pits in Los Angeles, suggested that direwolves were so different that they deserved a separate genus, but his view was ignored at the time. For the next 100 years, it was widely believed that direwolves were closely related to grey wolves and modern wolves. However, in 2021, a study published in the journal Nature completely changed our understanding of direwolves. Detailed fossil studies revealed that direwolves were very different from modern wolves. They had diverged from other wolf species 5.7 million years ago. The study also showed that direwolves coexisted with grey wolves and coyotes in North America, but there was no interbreeding between them, no gene mixing. About 13,000 years ago, direwolves went extinct, but grey wolves survived. The exact reason for their extinction is unknown, but it is believed that because direwolves preyed on large animals, and those prey animals also went extinct, the wolves too followed. Another theory suggests they went extinct because, unlike grey wolves, they couldn't interbreed and adapt to changing environments. Compared to modern wolves, direwolves were about 25% larger. Direwolves were portrayed as the size of lions, but in reality, they weren't that massive. However, their heads were definitely larger and broader, and their jaw muscles were very powerful. They had light-colored fur and lived in packs like modern wolves, making it easier to hunt other animals. Now comes the question of de-extinction. How did this company de-extinct a dire wolf? De-extinction means the reverse of extinction, bringing something back that had disappeared. Mainly, there are 3 methods for de-extinction. The first method is backbreeding. This involves selecting living relatives of an extinct species that still carry some of the old traits. Then, through selective breeding, the goal is to recreate those ancient traits. The quagga was an animal found in South Africa that went extinct in 1883 due to hunting. It looked like a zebra in the front, but like a horse at the back. Scientists selectively bred zebras with fewer stripes over many generations, and today, animals that look very similar to the quagga have been bred. However, critics argue that just looking like a quagga doesn't mean they truly are quaggas genetically. Cloning involves creating an exact genetic copy of an extinct animal using preserved cells. For cloning, you need a well-preserved cell of the extinct species. In de-extinction, cloning was first attempted in the year 2000 with a Spanish goat species called bucardo. Scientists cloned it, but the cloned baby died minutes after birth due to lung defects, making bucardo the only animal to go extinct twice. Though genetic engineering is considered the most modern and promising method, here scientists extract DNA from fossils, identify the genes of the extinct animal, compare them with the closest living relatives, and then edit the living species DNA to match the extinct species. Using CRISPR-Cas9 technology, for which Emmanuelle Charpentier and Jennifer Doudna won the Nobel Prize in Chemistry in the year 2020, they performed this highly advanced DNA editing. In 2021, billionaire Ben Lamb and renowned geneticist George Church founded the company Colossal Biosciences, their flagship project bringing back the woolly mammoth. They also aim to bring back the Tasmanian tiger and the dodo bird. However, all three species present major challenges like very long pregnancy periods for elephants or the difficulty of manipulating bird eggs. In 2023, the company decided that for testing de-extinction, the dire wolf would be ideal due to its popularity in pop culture. Think Game of Thrones, World of Warcraft, Dungeons and Dragons, and so on. By summer 2023, Colossal started work. They found two promising fossil samples, a 13,000-year-old tooth fragment from Sheridan Cave, Ohio, and a 70,000-year-old skull from American Falls Reservoir, Idaho. From these, they sequenced parts of the direwolf's DNA. They compared this with grey wolf DNA, which claimed 99.5% similarity. They chose 14 genes to modify, focusing on size, head shape, fur color, and fluffiness. Using CRISPR, they edited grey wolf cells. Then they grew thousands of these modified cells in a lab. In mid-2024, the next step was taken. They extracted the nucleus from these genetically edited cells. They inserted these into dog egg cells,